Hello friends, welcome back to my YouTube channel, The Microbiology Digest. I am Dr. D.P. Bora. Today we'll be discussing another topic of microbiology, another type of organism, that is Chlamydia organisms. The order Chlamydialis, the organisms under Chlamydia group. So we'll be discussing in detail about this group of organisms along with the disease caused by these organisms. Well, as I said, the order is the Chlamydiales. So these are also obligate intercellular bacteria. The organisms under this order Chlamydiales, they are also obligate intercellular bacteria, and they have unusual developmental cycle. These are uh, having some unusual developmental cycle. So the name Chlamydia, the word Chlamydia, it is actually given to this group of organism because of the characteristic appearance of their inclusion bodies. When these organisms infect the cells, they used to produce inclusion bodies, and these inclusion bodies they take a characteristic typical appearance. They appear like a cloak or mantle, and in actual meaning of the word chlamys is the mantle. So because of this, the name of these organisms, name of this group of organisms, it has been given as chlamydia. Now they are filterable agents, and they require living systems for multiplication, just like the rickettsial organisms. These organisms also they need a living system for multiplication. These group of organisms, another important character of these organisms is that they are generally not able to generate the energy compounds, and for this energy they are entirely dependent on the host cell. They are they are not able to generate the energy compounds by themselves, and for energy compounds they are entirely dependent on the host cells. So because of this characteristic, they are also sometimes called as energy parasites. The other name of the Chlamydia organisms is energy parasite. Now this group of organisms, Chlamydia, they exhibit some of the characters which are completely similar with the viruses, but they do differ from viruses uh, because they have a cell wall. They have a cell wall which resembles like gram-negative bacteria. They do not have the eclipse phase. In virus replication, eclipse phase is the period immediately after infection. This period starts, so that period is not present in case of chlamydia organisms. Then they produce intercytoplasmic basophilic inclusion bodies inside the infected cells, and sometimes because of this particular character they are also called as basophilic viruses, but not they are not exactly virus. And another character is that they possess both DNA as well as RNA. We all know that viruses, in case of viruses, only one type of genetic material is present. That is, it may be either DNA or RNA. But chlamydia organisms, they contain both DNA and RNA. They multiply by binary fission, and they are sensitive to antibiotics. So these properties, it they different these chlamydia organisms, they completely differs from the viruses. So because of this similarity as well as the dissimilarity between the viruses, they cannot be said exactly as viruses, and they are having some characters which can which are sufficient to differentiate them from viruses. So that is why it is said that chlamydia organisms, they occupy an intermediate position between virus and their other group of organisms, that is the rickettsial organisms. So they are having an intermediate position between virus and rickettsia. Now history says that the chlamydia existence of chlamydia organisms or chlamydial infections, they were recorded long back, but the actual work on chlamydia started with the work of Albert Neisser, von Proazek, and one more his of their colleague. They noticed some inclusions in the cytoplasm of the cells they collected from the scrapings of conjunctiva from a patient, and that patient was suffering from a disease called trachoma. So this was in the year 1907, and later on in the year 1929 to 30, three scientists, they could successfully isolate this or the organism responsible for this, and uh, three scientists, Levinthal, Cole, and Lilly, they were successful in isolating the organism. And after that, preliminary works on the disease psittacosis. This is a disease in birds caused by chlamydia organisms. It was carried by Samuel Batson, and in his honor, psittacosis agents are also sometimes called as Batsoni. Or in fact, the uh, genus name or family name Batsoni was proposed for these organisms, but later on now this chlamydia, the name chlamydia has been widely accepted. Now morphology, as we have already discussed, they are intracellular parasite. They are spherical, having unique developmental cycles. So this unique developmental cycle comprises of alternating two morphologically distinct forms. One form is called as elementary body, which is infectious form, and the other one is the reticulate body, which is the reproductive form of the organism. These elementary bodies, they are small, spherical, and infectious form, and also the extracellular form of the organism. And the reticulate body, which is the reproductive form, it produces by binary fission. After entering the elementary body to the cell, it gets converted to the reticulate body, and reticulate body starts multiplying by binary fission, and once it gets matured within the cell, it is again released from the cell so that neighboring cells can be infected. So this is the developmental cycle. Elementary body first enters to the cell, and after entering, it gets converted to reticulate body, and reticulate body starts multiplying, and finally again reticulate body, it is released from the cell. Now how we can identify this group of organisms? There are different type of staining procedures are available, and in different type of staining reactions they show different colors. So as they are gram negative, they can be demonstrated by chemical stains, or they can be demonstrated by fluorescent antibody technique, or using other serological techniques like immunoperoxidase technique. Then in modified Jell-Nielsen stain, they appear once we stain by using the modified Jell-Nielsen method. These elementary bodies, they tend to occur in clumps, and they stain red against a blue background. And similarly with other stains also, like methylene blue, then Machiavello stain, and Giemsa stain. These stains are commonly used for identification of this group of organisms. Now coming to the habitat, these organisms, in case of birds and mammals, GI tract is the natural habitat of these organisms, and these organisms can be shaded along with the fecal excretion, which is found to be a characteristics of these organisms. And the cetaceae, the etiological agent of cetacosis, it is commonly carried in the spleen and kidney of the birds. So affected birds, they can carry the organism in the spleen as well as in the kidney. Now, chlamydia organisms, they show a variety of resistance. They are heat labile and they can be inactivated within few minutes at 56 degrees centigrade. Then they are susceptible to ethanol, and they are also susceptible to low concentration of phenol and formalin. And their infectivity can be maintained at 4 degrees centigrade for several days. It has been shown that they can maintain their infectivity if it is stored at 4 degree centigrade. Then they can be, for long term, they can be preserved at lower temperature like minus 70 degrees centigrade, or even we can preserve the organisms by using lyophilization method. Then they are also, as we have already discussed, they are susceptible to antibiotics, like they shows high sensitivity to oxytetracycline, erythromycin, and psilocin. Next, coming to the cultivation part, cultivation, as we have already discussed, they are obligate intracellular parasite, so they need a living system for growth and multiplication. They are also called as energy parasites because they are dependent on the host cells for synthesizing the energy compounds like ATPs. So they can be cultivated by mouse inoculation. Then embryonic eggs can also be used, which is 6 to 7 days old embryonic eggs can be used. And the route of inoculation is mostly the yolk sac route. Then some cell lines also can be used, like McCoy cell line, BHK21 cell line, and Vero cell line. Even it has been demonstrated that these cells can be used for cultivation of this group of organism. Now coming to the pathogenesis. So as we have already discussed, the organism, the infectious form is the elementary body. It is the infectious form. So these organisms, they are released to the environment along with the fecal excretion of the carrier animals. Then these elementary bodies can also be shed to the environment through the semen of the infected animal. Now these elementary bodies are responsible for infecting others, healthy susceptible animals, and once it gets infected by inhalation or by through dust or droplets, these elementary bodies, they starts multiplying after entering to the host body. Then in case of suppose in case of EUs, it is responsible for some specific disease called as enzootic abortion. So here in this case, the organisms, they prefers to localize in the cells of placenta, and there will be invariably there will be abortion, and abortion, it occurs in the last 2 to 3 weeks of pregnancy. Again, in case of bovine, it is responsible for one specific disease called Bush disease. It is Bush disease, actually it is bovine unknown systemic sickness. This is also called as sporadic bovine encephalomyelitis, or SBE. This is a severe neurological disease, and it is caused by one species of chlamydia. It is Chlamydia pecorum. So here the vascular endothelium as well as the um, nerve cells are the site of cells where the organism prefers to multiply. Similarly, in case of cetacosis, as we have already discussed, spleen and the kidney, these are the organs where the organisms prefers to multiply. Now coming to the diseases, these group of organisms, they can infect variety of animal species including birds, and number of disease has been recorded to be caused by the Chlamydia organisms, for example Chlamydia psittaci. So if the infection it occurs in the psittacine birds like parrots, this is called as psittacosis. And if the same infection occurs in birds other than psittacine species, this is called as ornithosis. Then in case of other species of animals, it is responsible for some other diseases, like mostly the abortion. In many species it is associated with chlamydial abortion. In cattle, sheep, goats, in all the species it is responsible for chlamydial abortion. Then chlamydia pecorum we have already discussed, it is associated with a specific disease called sporadic bovine encephalomyelitis or Bush disease. Then in case of pigs, then in human mostly the in case of neonates, it is associated with a specific disease, it is called as trachoma. And because of this disease in infants, it is actually trachoma inclusion conjunctivitis. And because of this chlamydia trachomatis, and chlamydia trachomatis is also called as trick agents. Trick agents means trachoma inclusion conjunctivitis. This is the short form tricks agents. It indicates the chlamydia trachomatis. Now coming to the diagnosis part, chlamydia can be isolated from the blood during the early stages. It has been shown that, and specimens in case of abortion will have to collect the aborted material or aborted fetus and fetal cotyledons. We can demonstrate the can be used for demonstration of the organism. The uterine discharges we can collect, and in other cases, suppose for psittacosis, elementary bodies can be shown by direct smearing. Then we can go for isolation and cultivation. We can utilize serological tests like ELISA, CFT, etc. And we can also use polyacrylamide gel electrophoresis, restriction in the nucleus analysis for, as well as monoclonal antibody typing for further identification of the different species of the organism. Now coming to the treatment and control. So immunity against this organism is humoral as well as cellular level, and serotype specific antigens. It has been shown that they stimulates the production of antibodies, protective antibody. Then in some cases their vaccines are available, and vaccines can be used for prevention of this disease, control of this disease. And this organism, as we have discussed, they are susceptible to antibiotics, so we can go for tetracycline as well as chloramphenicol for treatment of this type of these infections caused by chlamydia organism. So this is all about actually chlamydia in short, and I hope these informations will be helpful for you. And if you find it is helpful, please subscribe to my channel and share with it your friends so that everyone will be benefited. And please put your comments so that I can further improve in our future discussions by incorporating more and more informations so that it will be helpful for you all. Thank you very much. Thank you. Bye bye.